This is the last video in the History of Genetics series. This video covers future topics that will deal with new findings in the genetics field. We have previously covered CRISPR in the History of Genetics Part 2. For a recap, CRISPR is a genome editing tool that has the capacity to remove, add, and alter sections of a DNA sequence. More specifically, CRISPR is a series of DNA sequences that are found in bacteria and archaea. They contain a protein, Cas9, that acts as a pair of molecular scissors that make it possible for the DNA sequences to be altered. This works because DNA sequences are detected from a DNA fragment from a prokaryote, a unicellular organism that lacks a membrane-bound nucleus, that already had a virus, and then CRISPR will detect viruses that are similar. There are many applications that could be possible for the use of CRISPR. Some of the areas that have been discussed and researched in this field are fighting cancer, eliminating infections, protecting plants, and in producing food. As we continue to see CRISPR advance, the potential for new possibilities that CRISPR could have are unimaginable. Another topic that needs to be considered for the future of genetics is the ethics that come about because of new scientific advancements. Currently, it is being argued that genome editing for reproductive purposes should not be attempted, but that the use of gene therapy should continue. Gene therapy is the delivery of a nucleic acid into a cell that is used as a drug to treat a disease. There are many safety concerns that accompany practices like genome editing. Some of the main worries are that the edits will be made in the wrong places, and that some cells could carry some of the possible edits, but some wouldn't. There is currently an effort between the US, UK, and China to regulate the use of gene editing technologies. Another worry that comes along with this is the issue of consent. The public is worried that there is a lack of consent because there is no possible way to obtain it from the germline editing. There is an argument that counters this, that parents already make decisions that will affect their future children, so what makes this decision any different? There also is a question of whether these practices will be available to everyone, or if this will just be available to the wealthy. This could cause future complications with access to healthcare and could create contention between classes of people based on the quality of their genomes. The ethics of certain genetic practices pose many questions, but the fields continue to push forward with new findings. The last topic that we would like to cover for the future of genetics is 3D printing. 3D printing organs is one of the newest methods that could potentially be used for different organ transplants. Scientists are able to construct a cell scaffolding to build a particular organ. This process begins with stem cells that are grown in a petri dish. These cells are then used as the ink cartridge to be fed into the 3D printer. From here, the printer arranges the cells with their different cell types, and a three-dimensional shape is produced. They haven't reached the point yet, but scientists hope that when this printed organ is introduced into the body, the cells will integrate with all other existing tissues. Bioprinting is accelerating quickly. In the next few years, it is expected to become a more common practice in the medical field. Genetics continues to progress, and the future of it holds many exciting possibilities that could have the potential to make substantial changes in the field of science.